Hi, this is Miss Litton, and this is AP Biology, Chapter 42, Review Animal Development. Say, hey. Hey. All right. Okay. So you had a whole lecture on this, which was so fun for you for that hour and 15 minutes lecture that you watched. But I'm going to do the Friday night highlight reel. Make sure you understand, and we'll do a little song with it as well. So real briefly, I know you saw this before, development, and the question is, to me, still amazing, how you get from a sperm and an egg to you, right? How many cells is that? 2 cells becoming 1. You were each 1 cell. And then look how amazing you are right now. So how did we get to here? From a single cell to a fertilized cell. So an egg will live in the oviduct for about 12 to 16 hours. Now you know a little bit more information because you understand the menstrual cycle. So when, what day of the menstrual cycle does ovulation occur? 14 days before the start, of your next cycle with the bleeding. Exactly. Yay, that's super fun. All right. So sperm needs to be present in order to facilitate that process. The sperm have a little cap, like a little beanie that they wear called the acrosome, and it has enzymes in it that helps penetrate the zona pellucida, which sounds like a summer concert band, girl band series or whatever. And so it digests through the zona pellucida. Now, once it fuses the sperm with an egg, there's a quick fix. Did I show the video in the sperm cam?
It was there.
It was there, okay. Sperm cam, that sounds terrible. So there's a quick fix barrier and then a long-term barrier. And you can see that here, the fast block, the sperm touches the egg. And just similar to the way an action potential travels on a neuron, and you're familiar with that. Yes? Okay. Same thing happens on the fast block to prevent any other sperm from entering the egg. And then a slow block is you have these vesicles right here, and they will secrete enzymes to make the zona pellucida just impenetrable by any other sperm. All right? That way you ensure that you have this. 1 egg fertilization is complete. The sperm and the egg nuclei have to join, and that's the fusion you see in the top right-hand corner. And then you have fertilization. Yay. Okay. So development is all the changes that occur in your entire life cycle. I'm in development because now I'm still what? What's happening to me? Oh, you're so kind.
Aging.
I'm aging. Thank you.
In a good way.
Okay, that was good. All right. Now, these are the stages I want to teach you. I want you to remember, if you remember from the lecture, that the steps of development and the movement of cells, which is called morphogenesis, when cells are moving about, that is going to vary dependent upon the amount of yolk, how much yolk is in the egg. So an egg that has a lot of yolk would be like a chicken, has a lot of yolk, okay? And then here what you're seeing is a lancelet in the top left-hand corner. I'm kind of pointing there. It doesn't have a lot of yolk, so the cells can move quite readily. But when you think about a chicken egg, the egg is actually, have we talked about what the actual egg is? It's a tiny little disc on top of the yolk. So you don't cleave through, you don't do mitosis through all of that yolk. And a froglet egg has intermediate amount of yolk, and so you kind of have to, the cells when they're moving about have to move around the yolk because you can't push the yolk aside. Does that make sense? So the dance of development will change depending on how much yolk you have. So the first thing that happens is I'll teach it to you here and then we'll look at it on the screen, okay? This is your egg. Here comes Mr. Happy Sperm, okay? What is Mr. Happy Sperm wearing on his head? Cap. What's the name of his cap? And what's inside his acrosome? Enzymes that digest zona pellucida. So that helps penetrate the egg, and then we form a diploid zygote. Okay? Then we do cleavage. Okay? And so basically what that means is it's doing mitosis so fast it doesn't have time for growth. It's doing the complete mitotic cycle within it, which relates just to the nucleus, right? But it doesn't have time to get bigger. So when it goes through cleavage, if this is how big your egg is, you have hundreds of cells in here and it's still this big because they're not growing. Does that make sense? Okay, cleavage, 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 and you become a solid ball of cells called a morula. Okay, so repeating mitosis, cleavage, a solid ball of cells called a morula. Okay, so now we're a solid ball of cells. Now, remember, we're going to blow this up like it's a water balloon, so we got to move water in. Remember, we can never actively transport water, but what's a way to move water? Salt, yeah. Put salts in the middle, so pump your salts in the middle, and then you're going to blow it up to a blastula. Blastula. And this is a hollow ball of cells. Okay, hollow ball of cells. Now, way I want you to think of that is like a red rubber playground ball. Like if you're going to play dodgeball. Have you got that? That's what it is. That is a blastula. And in humans, this happens on day 5. Day 5. Okay, that's a blastula. And you're still in the oviduct at this time. Okay, so from the top, here we go. Here comes Mr. Happy Sperm. His cap is called? And it contains to digest the zona pellucida. And then we form a diploid zygote. Cleavage, 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 and we're a solid ball of cells called a morula. Okay, then we pump in salt. Who follows? Water, and we're a blastula, and this is a hollow ball of cells. The cavity inside of here, okay, if we can see inside of it, it's called the blastocele. Say it. Blastocele. So I go blast and like seal your fingers together. Blastocele. Okay, that's what that is inside. Now, if the next stage, okay, the next stage I want to explain to you. And this would be a little bit different if we're human, so I'll come back and talk about the human aspect of it, okay? But let's just say we're a lancelet. What's going to happen is take that red rubber playground ball, and you let the gas out of that red rubber playground ball, and you're kind of like pushing your fist in like this, like so you could wear it like a hat, okay? You envisioning that? Okay? So you let out the gas, okay? And you become a gastrula. Gastrula. So let out the gas. What happens to the blastocele, this cavity inside? It's going what? Bye-bye. Bye-bye, blastocele. See how where it's gone now? Okay. Now it's called a gastrula. A gastrula is a layered ball of cells. Layers. What's the outermost layer? Ectoderm. What's the innermost layer? Innermost. Endo. Middle layer. Okay. Now, when you form a gastrula, initially there are just 2 layers, like red rubber playground ball. You have the outermost layer, which is the ectoderm, and the innermost layer, which is the endoderm, okay? Then the middle layer forms later. That depends if you are a protostome or a deuterostome, how that forms. I'm not holding you accountable for that, okay? Now, when you form this gastrula, form your gastrula, okay? The innermost cavity right here is called the archenteron. Say it. Archenteron. Okay? The archenteron, that innermost cavity in here, has an opening to it. That opening is called a blastopore. Say it. Blastopore. Now, if you are a protostome, like mollusks and spiders and bugs, that opening right there becomes your mouth. Okay, and then if you are a deuterostome like us, any kind of dermata, that opening becomes our anus. So if you have a complete digestive system, you have an indoor and you have a outdoor. This right here becomes our outdoor and a new mouth forms. Okay, for the other ones, it's their mouth and then an anus forms secondarily. So it's called the opening here is called our blastopore, and what I do is here, be right here, here's your blastula, okay, then we get infolding like this, invagination is what's happening right here, and then we form a layered ball of cells. First, just 2 layers. What are the 2 layers? Exoderm and endoderm, and then the mesoderm forms, and I'm going to talk about that layer. Okay, coming back here, this is just showing you 2 layers, right? When I do it like this, can you see the 2 layers? What's happened to the blastocele?
It's gone.
It's gone, okay? And now we have a new cavity called the archenteron. Think arc, okay? And like your thumbs make an A, archenteron. And then the opening to that, and you're going to take your elbow, go blastopore. Blastopore, because we're pointing to our anus, okay? Blastopore. Blastopore. Okay, good. Back to the layers. okay? Actually, let me go here. Okay, so here was the blastocell in the blastula, and then here's our gastrula, and our gastrula is a layer of all cells. Here's our archenteron, and then the opening right here is the blastopore. Now, if you are humans, inside the blastula, you will have what's called an inner cell mass, an ICM, inside of here. So here's our blastula. This could go be parts of the placenta, and then the actual baby comes from this inner cell mass in the middle. So that's just a variation on development. What I want to talk about now are those three layers. So if you look, the ectoderm is in blue, the endoderm is in yellow, and the mesoderm is in pink. And I need to talk to you about some significance of those layers. As far as what the ectoderm is, think about what's on the outside of you. What's on the outside of you? Skin. That's easy, right? Now, can you feel the skin right here? Okay. And what system allows me to feel my skin right here? So skin and the nervous system come from the ectoderm. That's easy, if you can just remember that. And if you can remember what the ecto and the endoderm are related to, then anything else has to be the middle layer, which is the mesoderm. Okay? So the ectoderm here is in blue, and I'll pull this out right here. Ectoderm, also matching in blue, exact color, because, yes. Epidermis, nervous system, and I'll talk to you about the lens of the eye in a minute. The endoderm, and remember the innermost cavity in here? What's that innermost cavity called? Archenteron. That is going to be your gut tube. So that's in yellow right here is your endoderm. Now, you want to think about what's related to your gut tube. So it can go in here in my gut and my digestive, and any glands related to my digestive system, that gut tube, that's going to come from my endoderm. But what else, what other system can I access through my mouth? My respiratory system. So my digestive system and my respiratory system are going to come from my endoderm. From my ectoderm is going to come my skin, and I can feel it, my nervous system. And then everything else, any other system you name, is going to come from your mesoderm. Okay? So your mesoderm has the remaining ones. Now, I bring up the lens of the eye, okay, because of this. If you look in this picture, this blue right here represents your spinal cord. Now, when you have those three layers and you start to form organogenesis, you're starting to form your organs, okay? That process is the ectoderm folds in like this, invagination, to form your spinal cord. Okay, so it was skin, it was on the outermost layer, ectoderm, but it folded in to form your spinal cord. Now, what got it to fold in to form your spinal cord was this pink thing right here, your notochord. Your notochord is a tissue that influences another tissue. It's like peer pressure, okay, where I say, put your hands on top of your head. Put your hands on top of your head, okay? So I'm making her put her hands on top of her head, okay? I'm influencing her. I'm influencing her with morphogens. I'm causing morphogenesis. Perfect. Okay. So the notochord gets the tissue above it to fold in and form a spinal cord. What they have done is some really creepy experiments, like with salamanders and stuff, and a few people. No, I'm just kidding. And what they did is they took like a salamander that was developing, and they removed some notochord tissue out of it and applied it to another salamander that was developing. So now it has two notochords. So it developed two spinal cords, two heads. Now, the same thing happens with your eye. You have something called your optic vesicle, which sits below your eye, has not formed yet, okay? And it induces your ectoderm above it to fold in to form your lens. Once the lens forms, it induces the optic vesicle to turn into an optic cup, which eventually has your rods and cones. So it's like this tissue says, you be this, and he goes, okay, then you be this, and okay. And so they influence each other. And that is all induction. They have repeated this with frogs, where they have taken optic vesicles from other developing frogs and put the optic vesicles along the legs of frogs, so they had eyes developing down their legs. Now, those eyes were not functional because they didn't have the extra optic nerves developing from the brain. Does that make sense? But the structures were there. They were just empty, dead eyes. Okay? So that leads to something further in morphogenesis, which is called induction. Now, the way I influenced her with words right here. Morphogenes is what really does it, a chemical. And guess what these really turn out to be? How can we turn genes on and off in mammals? What do we use? Eukaryotic cells.
Transcription factors.
Boom. They're transcription factors. So I influence her with transcription factors to turn on certain genes. Does that make sense? And that's how you can control development. All right? So, back to our development here. Okay, so gastrula. When you go to form the spinal cord, that is called a neurula, when that nervous system develops. And if you look, let me see right here. Oh, this is okay, let me step back with the gastrula. So this is showing you a lancelet, which is like that little clear fish-like thing I showed you. Then the second picture is a frog. Now, when we formed the gastrula, we just did rubber red playground ball, right? And let out the gas and folded it. And that's basically what a lancelet can do. A frog who has intermediate amount of yolk, they can't just pop in, fold like this to form layers. It has to grow up and around the yolk like this in order to form layers, because the yolk is going to influence that. And then last of all is the chick, and there's like a little blastodisc that sits on top of the egg in order to do it and forms layers. So I'm just telling you that the yolk is going to influence how the cells can move around, because it's going to limit that movement. Okay, now the neurula, I think I have, let me just jump forward, because I think I show you it. Yes, take a look right here. So this is an example of induction, but it shows you how you form the neurula. If you look up here, here's the notochord influencing the tissue of it above to fold in. There's a neural fold right here, and this neural plate, and then folds a little bit more, and then you have that neural tube on top, and that is an example of induction. Here they've got double spinal cords forming. Yeah, that's good. You got that. All right, and there's pictures in your book showing the different layers. So let's add to the story. We'll add just the very end. Here's an egg, sperm. Here comes Mr. Cap. No. Acrosome contains what? To digest the, and we form a diploid zygote. So the nuclei have to fuse between the sperm and the egg, yes? Then we do what? Cleavage, 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 and we form a solid ball of cells called a morula. We pump in salt. Who follows? Water. And we form a blastula. And a blastula is a hollow ball of cells. And we form that on day 5 for you and I. Good. And what do you call the cavity inside? Blastocele. What's the next stage we're going to form? Okay, and it's forming a gastrula. How a gastrula form is dependent upon the amount of yolk. Okay. So we're going to form a gastrula in the simplest way, and at first we form how many layers? 2. What's the outermost layer? Ecto. And the innermost layer? Endo. And then the middle layer. Now let's talk about that. From the ectoderm you get your skin and nervous system and lens of the eye. From your endoderm you get your yeah, so it's that inner tube and any associated organs and glands, which would be our digestive system and our respiratory system. And everything else comes from the mesoderm. Okay? Now, when we form that gastrula, what's the new cavity called? Archenteron. And then the opening to that new cavity? Blastopore. Good. Now, the last stage we form is a neurula. And to represent the neurula, I just push my fingernails together. And that's the invagination to form the spinal cord. See what I'm doing? Okay, that's a neurula. All right, perfect. Okay, do you want to answer some questions?
Yes.
Okay,
yes, yeah.
Here we go. Let's go in as our favorite baby animal.
Not a pig.
No.
I'm so excited. I'm not confused.
Wait, we're digesting
Well, last time you had bacon, you did. Okay, I'm going to pause you while they answer the questions. Okay, here we go. Yeah, so if you put that in order, it would be cleavage, morula, blastula, gastrula, neurula. Number 2, guys, I don't have time. Which one is mismatched? Blastula for gut formation. What stage would you need to put there for gut formation? Gastral. right? Okay, so the notochord, I forgot to mention this. The notochord in most mammals, subphylum Vertebrata, would be, the notochord becomes the bone around the spinal cord to protect it. The notochord itself, what induced it, becomes the actual bone. And archenteron will develop into the gut. And that's the end of that. Okay, so continuing on. Cellular differentiation. When cells differentiate, they become specialized. Morphogenesis is the movement, the process, the dance of development. And apoptosis, you know what that is. Apoptosis is a normal part of development, right? Because if you look at your hand would be a paddle unless what? Apoptosis occurred between your fingers. Radius, which comes up from your thumb, and your ulna right here. If the cells didn't die in between it, then you would never be able to turn your hand over, right? You could only do it at your shoulder. The reason why you can rotate your thumb over your ulna like this is because the cells died in between that to allow that kind of rotational movement. Otherwise, you would just be like this all the time. Okay? So apoptosis is a normal part of differentiation. All right. So now, here's the kicker. Okay? Every cell, when it's first formed, sperm, egg, diploid, what? Zygote. This is called, it has complete totipotency. If you are totipotent, you have full developmental potential. For us, you can divide once, twice, you can be up to 8 cells, okay? And still, every cell would have complete totipotency. So if you had an 8-cell baby, okay, and you took it out and you divided those 8 cells, you could make 8 identical octuplets from those cells because they have complete totipotency. But if, let's say you get to 16 and 32 and you try to divide it in half, it's not going to develop. Bad things are going to happen. Some organisms lose totipotency following the very first division. They lose it. Cells are already predetermined in what they're going to be. Okay, so it varies per animal having complete developmental potential. If I took a skin cell, I couldn't take a skin cell off of her and implant it in my uterus and then have a baby, right? Those cells are specialized, they've differentiated, they don't have the ability to make another whole baby. Does that make sense to you? Okay, just letting you gross this out. Okay, now, let's talk about that, okay? The DNA in every single cell in your body is exactly the same. Do you agree with that statement?
Yes.
Okay. So it's like, how do they lose totipotency? What's going on? Well, it's the cytoplasmic constituents. What's in the cytoplasm? And the best example to show you that is to do a drawing, which I probably did in the lecture as well. Did I draw a circle like this? Do you remember this? Yeah. And at the bottom of this circle, right here, would be yolk.
Oh, yeah.
Did this?
Yeah.
Okay. And here comes Mr. Happy Sperm, right? So now we have 4 quadrants. Okay, this one has no yolk, and it does have sperm. This one has what? Yolk and sperm. This one has yolk and no sperm. Thank you. And this one has
no sperm, no yolk.
No sperm, no yolk. Okay, so when you start to divide these cells out, the cytoplasmic constituents are slightly different. Those things can impact what DNA gets transcribed and translated. You know them as
Transcription factors.
Exactly. So you have subtle differences, okay? If you touch the top of your head right here, touch the top of your head, that is where your father's sperm penetrated your mother's egg. It becomes the anterior region. So there were differences in the chemicals. Go home and ask your dad. Look familiar. Okay? So, maybe they won't appreciate that. I don't know. So there are subtle differences in the chemicals that the sperm brings in that area that you don't have on the other end. So that's your anterior, posterior. And then you have your ventral dorsal differences as well. Right here, tap your belly. This is what's called the vegetal pole, not vegetable. Think like, if I eat vegetables, I don't know. Ventral pole is where the yolk is. No yolk is dorsal. So if I have sperm, it sounds terrible. I've never put it together this way. Sperm and yolk will be your face. Sperm and no yolk will be the back of your head. Sperm and yolk will be your, or some, this is halfway between. So it's like, right? It's gradients. And those gradients, okay, there's gonna, whoa.
Oh.
Right? So it's incremental, right? And this is how you get differentiation. This is how you get segmentation, right? Because this would be a lot, a little less, a little less, a little none, right? A lot, a little less, none. So now I've got a gradient of two different chemicals. Do you agree with me?
Yes.
Every plot point I pick on here is different. Yes?
Yes.
These 3 plot points as far as segmentation goes for anterior, posterior, dorsal, and ventral. There's slight differentiation between that. These all have, let's say, the same amount of sperm, but they have variation in sperm and the amount of yolk. Do you agree with that?
Yes.
So this is what creates those patterns in our bodies. So, for instance, if you look at a frog, the back of the gray frog, there's an area, it looks like kind of an upside down smile called a gray crescent. And what they did is they took some experiments where they forced a developing embryo apart, one where each got the gray crescent, and one where one got the gray crescent and one got none. And this is as far as it got. It never moved into the gastrulus stage because what they realized is where the gray crescent is where gastrulation begins. It's the dorsal lip of the blastopore, and it folds in right there. And so if you don't have a gray crescent, you just turn into a ball of cells. So that is key during development, these little slight differences. And the gray crescent shows up on the opposite side of where sperm penetration takes place. When you talk about full developmental potential, that would be if you are totipotent. Okay. Do you see that I'm a pointer? Okay. So that morula gives rise to all the cells that make up the embryo and all the support tissues as well. Okay. Now this right here in humans would be called the inner cell. Oh, here it is, right here. Inner cell mass. Do you see that? That's what actually develops into a human for us. If you are pluripotent, it gives rise to the cells of just the embryo, where the other ones are going to give rise to cells for attachment or the placenta. And then unipotent, what does uni mean? One. It means you have started down a developmental line and you cannot change gears and de-differentiate and become something else. That's why when we cut off hopefully, but if you cut off your arm, it's not like I'm going to have a little bit of an arm, I'm like, oh, that arm's only like 2 years old, looks so cute, okay? It won't grow back, right? Like lizards do. We don't do any de-differentiations. We're not like, what was that movie? Deadpool. Yeah, we don't Deadpool, okay? And so it's become too specialized at that point. Okay. We know Deadpool.
And have the healing too.
Oh, wouldn't that be awesome? Okay, I already said induction. I talked about the frog eye. I've talked about that. Okay, so they could ask you something about C. elegans. It is a worm. It has been studied quite a bit. It's 1 millimeter long. It has a total of 959 cells, and it takes three days to develop. And they've done a lot of research on it, and they've looked to see, like, they know this will become this, and this cell will become this. They know it's already, there's a difference between being determined and being differentiated. Determined, you might be determined to be a doctor, and you're going and getting ready to go to school in order to be that, but you aren't a doctor yet, right? So when you get differentiated is when you become specialized to that. Could you change your mind if you're determined? Possibly. It depends on how far along in development. They've done some pretty interesting experiments where they've taken where like wings have not developed yet, but they moved tissue that is determined to be a wing, and they've added it, and so they ended up making double flies because that tissue was already going to develop into it. It just hadn't done it yet. So they were able to add it in there and then get 2 wings. Okay, I'm going to go back here. Morphogenesis. Morphogenesis is the movement. Morph is to move. Genesis is the beginning of movement. Is the movement of cells that changes the shape and form of body parts. And then this is what I talked to you a little bit about before about induction. And how I induce you is with morphogens. So as a result of me saying to her, put your hands on your head, put your hands on your head, okay? Now she's turning on these genes, which then makes a neighboring cell be something else, which makes a neighboring cell be something else. And so it keeps specializing, and it does that ultimately usually with the homeobox genes, which are transcription factors. Do you remember that from the presentation a little bit? Okay. And this is how we get segmentation. And so the specific features develop at each area. It's not like I have arms here, and then it's not like you see people that have arms coming out of their hips, right? I mean, I don't know that I've ever seen something like that. Now, are there ever cases where somebody might have multiple? Yes, there are. That's when things are not working according to plan. That would not be a normal thing that happened there. Some extra genes were turned on because you would have the ability to have another set of arms come out of here. We just don't turn those genes on. So there's been a problem with the communication. So do you remember when I talked to you about where I overlapped the amount of yolk and I overlapped where the sperm is and those slight differences? Then those lead to other differences which trigger other genes, which trigger other genes, and it becomes more and more specialized. Okay, so if you look, you have what you got with the mother, just the straight up yolk, and then the changes that are applied with the sperm. Okay, what else do I want to show you? I showed you a little bit about this already. It ultimately affects morphogens, and morphogens are transcription factors. You saw this in the lecture. Hox genes. When you look, here's a fruit fly, a mouse developing and a fruit fly developing, and you can look to see how similar the genes are. So it's going to be a head, just what kind of head is it going to be? That pattern is repeated over and over again. And so those genes are conserved, right? And that can also lead to evidence to support the idea of what? My name is Luca. There's a song. Last universal common ancestor, yeah. All right. Okay, I have more questions there, but I think that is the end of it. Yeah, because this is baby, and you don't need to know that. So here, if you want to do these questions real quick, I'm going to pause you. Okay, so let's finish this up, just because I have an appointment when the bell ring. Good job, two of you. Yeah, morphogenesis is associated with all of those things. Protein gradients, induction, transcription factors, and homeotic genes. Okay, it's connected with all of those. Number 2, which of the following is mismatched? Good. Lens from endoderm, how would you change that? Lens from what? Ectoderm. Good job. And number 3 is ability of one tissue to influence another tissue is called induction. Okay? All right, be smart, make good choices. I will see you tomorrow. If you're tired, have a piece of toast.